think we can start, Darcy.
Okay, so but can you reshare? It just says I'm viewing, so I don't think that I can control your screen right now. No, I can. Perfect. Okay. Yep, great. Okay. So welcome, everybody. It looks like we still have some people in, but we have over 50 people here. So welcome tonight or this morning, depending where you are in the world, webinar. My name is Darcy Wagner, and I am the current secretary for the ERS Group 3.03, which is basic and translational science for mechanisms of lung injury and repair. And amongst the members of the Assembly 3, we decided to initiate this webinar today to really talk about the different models and tools for COVID-19 research, especially what's emerging in the basic science area. And so we're very fortunate enough today to have two excellent speakers who have published some of the seminal work so far in this area. So Stefan Pullman and Josef Penninger. And so the format of today's talk, and actually really quick, I have no conflicts of interest of major, but I've received speaker fees from Bayer Engelheim. But the main aims of today's webinar is to really go over the mechanisms of SARS-CoV-2 cell entry and infection, and also to hopefully facilitate at the end of the two speakers really a discussion about the different models, in vitro models that can be used to study SARS-CoV-2 infection as well as COVID-19 disease. And then also to have a discussion about the available tools to visualize and map sort of the infections along the respiratory tract. So again, the format today is that each speaker will have 20 minutes, and then I will moderate a discussion at the end. And hopefully most people here by now, because we're far enough along in this pandemic, that you are well acquainted with Zoom. But for this webinar, we'll be using the chat feature in order to ask questions. So I will moderate those. So please type all your questions in the chat box and make sure that you have it selected so that it goes to participants or attendees and panelists so that I can see that. So just from a real basic perspective, for those of you that maybe are not following all of the latest developments, I thought I would just provide sort of a comprehensive overview just some basic information. So there's been several different papers that have really sort of delineated, again, we're going to listen to the first speaker about ACE2 expression and how that receptor is critical for entry, but it's also important for us to understand from the respiratory side of where that receptor is expressed. And so this is work that was published in from Hu et al. In Cell recently, and what they did is they characterized sort of where ACE2 expression is along the respiratory tract, and what they found was that there was very high expression in the nose and in the bronchial region, and sort of that sort of diminished along the respiratory tract, but they were able to find cells infected sort of all along the respiratory tract. There's also been several different single cell data sets which have been leveraged to sort of ask this same question. So not looking at tissue staining, but really data mining. And this is up here for anybody else to also explore. These have really great user interfaces. So as sort of this research progresses, if you want to see what cell type, sort of a gene that you're interested in, if it's expressed, you can access these. And these are huge consortiums that the respiratory community really, and great resources from several different very prominent groups that have made those resources available. So again, just a reminder to everybody that, again, I just showed you that the highest, infectivity and also expression of ACE2 is in the upper airways, but this is actually not why patients are dying. So patients, if it's respiratory failure, it's respiratory insufficiency, and this is happening in the distal regions. So not necessarily correlating with high levels of the receptors for viral intake. We also know by now that there's major comorbidities and that underline this, as well as additional causes of death. And this is cardiovascular, including stroke and arrhythmia, as well as diabetes and renal failure. With regards to established models that already exist right now, so both of the speakers today are going to talk about sort of their use of different models. But with regard to the lung, there are several different models, and this depends on what area of sort of the lung you want to target. So if you're more interested in really looking at the upper airway epithelium, there's several different options. So of course, there's primary airway or nasal epithelial cell culture. These are then lifted to air-liquid interface to create differentiated epithelium, because what much of the data from both histological staining, immunohistochemistry, as well as single cell data indicates is that it's maybe more prevalent in sort of the differentiated epithelial cells. There's also ongoing work in organoids, which we are going to hear a little bit more about today, both from primary or IPS derived. There's also airway on a chip. And then the same thing goes for different mono or co-cultures that can be applied for studying the distal epithelium. Again, organoids, either primary or IPS derived, as well as precision cut lung slices, and then alveoli on a chip. Again, there's different ways that you can think about this. I apologize that this has shifted for some reason. But again, these are sort of the primary models, and many of them have different readouts that have been well established by now. So you can look at things like gene and protein expression changes in these in vitro models. There's quite sophisticated imaging techniques as well that have been developed for them. Really, there's a variety of different models that exist for studying COVID-19 or SARS-CoV-2 entry that span all the way from synthetic biology. So if you were interested in really looking at receptor interactions, there's a lot of potential tools with synthetic biology, monoculture, differentiated culture, and you can really see that, depending on your research question, that these build with increasing biological complexity. And then if we go to really sort of the things that are very interesting of looking at sort of the tissue or the systems level, there's a couple of different options, being lung tissue explants or precision cut lung slices, perhaps serial connections of different organs on a chip. And then also that currently right now, at least the literature that I was looking at to prepare for this, that there is evidence of Syrian hamsters actually being infected, that they can be infected, and that they're a decent model, that they mimic some of the features seen clinically. So again, depending on where your research question falls, there's different models that may be available for your research. And again, just sort of to set the tone here at the beginning, I think in addition to us sort of, understanding what's happening right now, that as a respiratory community, I think that we should already start to think about what new challenges will these infected patients face in the future. Will there be emergence of any new respiratory diseases? Again, we're going to have from the speakers today also discussion about effects on other organs. And it's really a question then of what models do we have to study this. So with that, I will turn it over to Stefan Pullman, our first speaker, to talk about SARS-CoV-2 entry.
Thank you very much. Let me just share my screen. That did hopefully work, did it? Yes, I think that worked fine. So thank you very much for the kind introduction, and thank you very much for the possibility to present my data. The title of my talk is SARS-CoV-2 entry into cells and its inhibition. The idea is to give you a brief overview over the coronavirus zoonosis first, then introduce the major topic of my presentation, the coronavirus spike protein, which is the key to the host cell, and then talk about proteolytic activation of the viral spike protein and its inhibition. So coronaviruses, I can be very brief. Those are enveloped RNA viruses that have several membrane proteins, and the most prominent ones is the spike protein. You can actually see it in the electron microscope, and it provides the virus particles with a crown-like corona shape, and this is what coined the name for this family of viruses. It's not unfair to state with kind of a smile that in the 1990s, coronaviruses were not really a hot topic. And this is why. This is because those viruses which have been transmitted from animals to humans. Sorry, let me start different. The reason is that there are human coronaviruses that have been a while ago transmitted from animals to humans. now exclusively circulate in human, and this is the major point, they do not cause any kind of severe disease. They cause common cold-like symptoms, and therefore they were not in the focus of scientific research. However, as you all know, this has changed dramatically during the last decades, because it was realized the hard way that some of those animal coronaviruses can be zoonotically transmitted to humans, and this has dramatic consequences. This has first been noted in 2002 when the SARS coronavirus, or SARS coronavirus 1, jumped from animals, likely civet cats, to humans and caused the SARS epidemic. Next time that happened was with MERS coronavirus, now in the Saudi Arabian Peninsula. This virus is transmitted from camels to humans, and like the SARS coronavirus, causes a severe respiratory disease that is called MERS. One commonality between SARS and MERS is that the viruses causing the diseases are not very efficiently transmitted between humans. Unfortunately, this is not the case for the third virus that made it from animals to humans. That's the SARS coronavirus 2 that is responsible for the current COVID-19 pandemic. And I don't have to say much here. The pandemic is associated with horrific numbers and has dramatic medical, social, and economic consequences. So let's look at more detail at the virus that causes the pandemic, specifically at the coronavirus spike protein. So this is the basic concept. It's simple enough. The spike protein sits on the surface of the virus and mediates virus entry into target cells. And my labs and others could show that for this, it needs to interact with host cell receptors, specifically the ACE2 protein, which is also used by SARS coronavirus 1 as entry receptor, for access to target cells. So if you want to, the spike protein is the viral key to the target cell, and the ACE2 protein is the access door. The point, however, is that the key is not just working, it needs to be activated, and for this, it needs to be cleaved by host cell proteases. Now, let's look at the S protein in more detail at a molecular level and look at the domains that are important. For one, the S protein is subdivided into two subunits called S1 and S2, and they have very distinct functions. The S1 subunit mediates or contains the receptor binding domain, short RBD, that mediates the interactions with ACE2. And then there's the S2 subunit that mediates fusion of the viral membrane with a host cell membrane. And for this, it needs to be cleaved by host cell proteases. And this cleavage event occurs at the S1, S2 site and the S2 prime site. And importantly, this cleavage is essential for viral infectivity, and therefore the responsible cellular proteases are potential targets for antiviral intervention. And it's also noteworthy that the two sequences that we just talked about, the ACE2 binding site and the cleavage site, were early on recognized as very particular. They were unexpected, and they even sparked discussions whether SARS-CoV-2 could be a virus generated in the laboratory, designed in silico. And of course, now we know that this is not the case, and we know this because related viruses have been identified in animals, in pangolins and bats, and turns out that some of those viruses harbor those unusual sequence elements. And this is very clear evidence that SARS coronavirus 2 is of zoonotic origin, has probably jumped from animals to humans within wet markets in China. So let's look at more detail at proteolytic activation of the viral spike protein and its inhibition. And here I would like to introduce work conducted by Hanna Kleine-Weber and Markus Hoffmann in my laboratory. What did they do? They first generated a model system that allows to study SARS coronavirus 2 entry in the BSL-2 laboratory because it relies on pseudotypes based on the vesicular stomatitis virus. It's very safe, it's very fast, and it's highly quantitative. And we distributed those reagents to many laboratories worldwide and hopefully help to initiate some interesting projects. And this is one example of what you can do with this system. We used it jointly with colleagues in the US and in Belgium to study antibody-mediated neutralization. And those studies revealed that particularly antibodies, so-called single-domain antibodies made from llamas, can do a very good job at inhibiting SARS-CoV spike-driven entry. Passive immunization is, of course, the ultimate goal of that research. In terms of activation, we looked at this process in some detail, and basically all our findings are summarized in that slide. We obtained evidence that proteolytic activation of the SARS-CoV-2 spike protein requires 2 cleavage events. First, the spike protein needs to be cleaved by the host cell protease furin in infected cells, and cleavage occurs during transport of the spike through the constitutive secretory pathway, and it occurs at the S1, S2 site. That cleavage is absolutely essential for a second cleavage event, now occurring at the S2 prime site. And this cleavage event, which occurs during entry into lung cells, is carried out by a cellular serine protease with the unattractive name of TMPRSS2. And the dogma is that this is also absolutely essential for infection of lung cells in the host. There's a third activation pathway that I will talk about later, but the current belief is that this is only relevant in cell culture. So let me provide you with some primary data. What did we do to identify the two-step cleavage mechanism? Well, for one, we mutated the S1, S2 site, and then we looked at 2 different commonly used cell lines, Vero cells, which are TMPRSS2 negative, Calos3 cells, which are TMPRSS2 positive. And the simple message is that if we destroy or alter the S1, S2 cleavage site, we do not see a big effect in Vero cells because they can use this alternative, or here for entry into those cells, this alternative pathway can be used. And for this alternative pathway, S1, S2 is not important. Very different in Calos3 cells. If you destroy the S1, S2 site, entry basically sinks to background levels. So S1, S2 is really important in our system. It's not really important in our system, but also in other systems. Many researchers find that if you grow SARS-CoV-2 in Vero cells, it loses its S1, S2 site because it uses this alternative activation pathway. And now if you take this virus, put it into lungs, you see that it is much less able to grow and is less able to make animals sick. And for these studies, the hamster model was used, clearly providing evidence that an intact S1, S2 site is required for SARS-CoV-2 spread and pathogenesis in vivo. Now, how about the second cleavage event carried out by TMPRSS2? Here we first used pseudotypes and we used an inhibitor, camostat mesylate, of which we knew that it would inhibit TMPRSS2. And indeed, we found that this inhibitor targets MERS, SARS, and SARS-2 spike-driven entry, but has no effect on entry driven by a heterologous glycoprotein, the G protein of VSV. And importantly, those findings made with pseudotypes could be confirmed with authentic SARS coronavirus 2. Here, infection of lung cells and its inhibition by camostat is shown. So what is the relevance here regarding treatment options? We feel those findings are very relevant because camostat has been approved for human treatment in Japan. It's known to be very well tolerated and is currently being assessed in several clinical studies. And the first one that has started takes place at Aarhus University Hospital in Denmark. So in short, Camostat mesylate joins the drugs that are currently used or explored for SARS coronavirus 2 treatment, including remdesivir and chloroquine. Is there any evidence that Camostat may do anything good in COVID-19 patients? We examined this. Camostat was used within compassionate use at the University Hospital in Göttingen, and a small group of 5 patients was compared that received treatment with either hydroxychloroquine and Camostat mesylate, and you can appreciate that expression of inflammatory markers goes down when Camostat mesylate is used for treatment, but not when hydroxychloroquine is used. And also, if organ failure is assessed using the sequential organ failure assessment SOFA score, we see that patients on Camostat mesylate do, in this case, even significantly better as compared to patients treated with hydroxychloroquine. And to finish this argumentation, I would also like to point out that there's now an even better inhibitor. It's a related name. It's called nafamostat mesylate. And if you look at the IC50, compare the second line and the third line, you can appreciate that nafamostat mesylate is doing even significantly better than Camostat mesylate. And finally, I would like to set this into the context of currently used antivirals. There's kind of agreement that remdesivir is working. It's blocking the coronavirus polymerase, and there's at least one large clinical trial indicating that it reduces time of hospitalization. Doesn't protect from death, at least not in this trial, but it reduces time of hospitalization. Then there's the famous chloroquine, which unfortunately became a strong political angle. And we looked at that, and it turns out that in our hand, chloroquine blocks this cell culture only pathway, the cell culture artifact, does so very efficiently, and therefore infection of vero cells, which are African green monkey cells, can be nicely blocked by the compound. But if you look at lung cells, you don't see any inhibition, meaning that we do not expect chloroquine or hydroxychloroquine to work in patients. Then, of course, there are other applications, but they will not be discussed today. In fact, at this point, I would like to summarize. I told you that the SARS coronavirus 2 spike protein mediates viral entry, that the S1S2 site is cleaved by furin in infected cells, and that cleavage is essential for subsequent viral entry into lung cells. This subsequent viral entry into lung cells depends on TMPRSS2, which activates the spike protein. Finally, there's inhibitors, Camostat mesylate and nafamostat mesylate, that block TMPRSS2 and inhibit SARS-CoV-2 infection of lung cells. And finally, there's first evidence that Camostat mesylate may be suitable for COVID-19 treatment. And I realized there's a preprint out of Japan that also reports that nafamostat mesylate is doing a good job in COVID-19 patients. With this, I would like to thank the people who did the work, very particularly Markus Hoffmann and Hannah Kleine-Weber, but also the lab of Christian Drosten at Charité in Berlin, and lab of Martin Winkler at the UMG in Göttingen, BMBF for funding, and you for your attention. And I should declare I'm a bit embarrassed because I filled out the COI form only seconds or basically minutes before the presentation started. I did receive speaker fees from Novartis and a honorarium for Merck for consulting. I apologize for not putting that on the slide. Otherwise, I don't have a conflict of interest. Thank you.
Thank you very much, Stefan. And we'll take questions for those who have joined later. We will take questions with both speakers at the end. So now I would invite Joseph. I think Ali will get the screen switched.
Yeah, pleasure to meet you all.
Joseph can share the screen right away
Okay,
and his presentation.
let's see. Here we go. Good. So thanks so much for inviting me and to follow after Stefan. Brilliant talk, and of course brilliant and really seminal research in coronaviruses and how they enter the cells, and of course leading to important therapies. So I want to tell you a little bit about our ACE2 story, how we came about to work on ACE2, and how we recently working on human organoids to study infection. So that's my conflict of interest. I started a company called Apeiron. I'm still a board member, so I have shares, and actually in phase 2 clinical trials with soluble ACE2.
Joseph, sorry, your microphone is going down a little bit from time to time. Can you select another one, or
no, that's my only.
Or approach a little bit your computer?
Yeah, I will try to speak louder. Okay, good. So we actually started this when it was 1998. I was interested in heart development of flies. There's actually P elements in flies, and we were interested in finding genes which would control this. And then we realized that actually flies had 2 orthologs of ACE. And so as a postdoc in my lab, Mike Krakauer cloned the second ACE, which of course was then called ACE2. I should also mention we were not the first group which published the sequence. This was Tony Turner and a group in the US. We already had the sequence in our freezers. The reason is because I'm a functional genetics guy, so I make knockout mice. And we really wanted to understand the function of ACE2 in a living organism. So, but as you can see, ACE2 is this transmembrane molecule, and it has very high homology to ACE. So this was the first knockout mouse which existed, which we published in Nature 2002. And the first phenotype, which I, in retrospect, still find very interesting, was actually in this particular mouse background, we saw impaired heart function, which I think is quite interesting considering COVID-19 now. And over the years from my group and many other groups, and in particular, this in vivo knockout experiment showed that ACE2 is actually the more or less the negative regulator of the renin angiotensin system. In essence, ACE, the classic enzyme which had been known for decades, is cleaving 2 amino acids from angiotensin 1 to make angiotensin 2, an octopeptide acting on 2 G protein coupled receptors. And ACE2 cleaves the last amino acid, at least in biochemical assays, it can do much more, but in vivo, this is the critical function, cleaving the last amino acid of angiotensin 2, and by doing so, inactivating Ang2. And of course, all the consequences we, and of course, many others in thousands of papers have described in terms of blood pressure, heart function, kidney disease, fibrotic diseases, diabetes, you name it. Of course, where all the ACE inhibitors and the inhibitors of the AT1 receptors come in. I should also point out that ACE2 is not specific, so it also cleaves the terminal amino acid from other systems like apelin, which regulates angiogenesis, bradykinin, which regulates leaky blood vessels. So this is where we were. And then we realized ACE2 is also expressed in the lung. And we looked for normal lung function. And when you actually knock out ACE2, we did not see any difference. So in normal physiology, ACE2 does not appear to be important, or the renin angiotensin system, for the lung functions, at least the ones we can read out. So a postdoc in my lab, she has now her own lab and is a professor in Japan, Yumiko Imai, developed an intensive care unit for mice. So at this time, very few people actually worked on this because it was very difficult to standardize experimental protocols for reproducible lung injury models. And so Yumiko worked for many years also with Atzlatsky in Toronto to actually set up this model. So this was our initial model, acid aspiration injury. As you can see, we change in elastance, so lung function. There's edema formation after acute acid challenge of the mice. There's inflammation determined by MPO activity. And this is how the histology looks. So the hyaline membranes, the edema formation, there's inflammation. Very rapid, happening within hours after we challenged animals. So we use this model to basically study the function of ACE2 in acute lung injury. And these were our data, which really surprised us. So on the top, there's already lung injury, excuse me, in a normal mouse. We inject Evans blue to look for leaky blood vessels. You can tell it's already a little blue here. Excuse me again. And when we knock out ACE2, you see severe acute lung injury and the blood vessels just start to leak. So this is where we were. And then, of course, as Stefan already pointed out, SARS came among the world. I was just moving from Toronto back to Europe at this time. And of course, just to put it in context, SARS infected around 8,000 people globally. And it took around 10 months to actually kind of control this. So with COVID-19, we are in the tens of millions probably of infected people. So the idea that, you know, this will just miraculously disappear will unfortunately not pan out. If with 8,000 patients, it took even nearly 1 year to control this. Also, it was interesting to live in a city which was the epicenter of SARS outbreak outside of China in Toronto. And when I was there, it was quite interesting because I think at the end there were around 350 people infected in Canada, in particular in Toronto. But this completely shut down the economy, movie industry had to stop. Lots of the movies were made at this time in Toronto. So it was actually really interesting how a little virus can completely disturb the social and economic fabric of a 5 million people city. And then we were surprised by this paper from Mike Fasan in Cell Lines, where he actually published that the coronavirus would use ACE2 as its entry point. And Stefan and Darcy have already pointed this out. So, but initially, there were other receptors proposed, in particular lectins, because the spike proteins are heavily glycosylated. So the question was really, is ACE2 the essential receptor for the SARS coronavirus in vivo? Since we had the only ACE2 knockout mouse at this time, so we sent it immediately to China. They infected it with the mouse-adapted SARS coronavirus. And when the experiment came back, we can recover virus in wild-type mice, but in ACE2 knockout mice, there's basically no virus, and also no pathology. It's mild pathology, but it was totally gone. So basically, this experiment was proving in vivo, in an animal model, that ACE2 is the critical receptor for the SARS coronavirus. And over the years, and with our work, and of course work from others, we came up with this idea that actually, and this became also now a center of attention for COVID-19, that lung injury, acid aspiration, pneumonia, sepsis, turning on the renin angiotensin system, which was at this time really a new function of the system in the lung. And this triggers Ang2 production, and via the AT1 receptor, because we do all the genetic mapping, the blood vessels open, there's severe lung injury. So basically this pathway gets turned on and contributes to the severity of lung injury. ACE2 is the good guy in the system, so because it gets rid of Ang2, and of course if we knock out ACE2, then the disease gets much worse. Now the virus has basically occupied ACE2, it uses it to enter the cells, and by doing so it downregulates ACE2 expression from the cell surface, and one more or less ends up with an ACE2 knockout mouse. And we believe this is one of the mechanisms and one of the molecular explanations by the SARS virus, and we also believe the second SARS virus now causing COVID-19 became such a severe virus because and affecting multiple tissues because ACE2 is actually protecting tissues from injury. In this case, the lung, it also protects other tissues from injury, like the kidney, the blood vessels. We and others have published over the years, and of course the heart, as I showed you. So based on this, we showed if this is actually reduced ACE2 enzyme activity, we could make a soluble form of human recombinant ACE2 and put it in our model. And as you can see, there's acid injury, and if we infuse this molecule, we can stabilize the acute lung injury. So over many years in this biotech I mentioned, it just started, and again, declared my conflict of interest, so you can put it in context. We actually developed this molecule. Of course, then SARS disappeared. The interest waned, was considered a very nice finding, which we did. But the question was really, who cares? Because, of course, there was no SARS anymore. MERS uses a different receptor. And so we developed this molecule for ARDS, acute respiratory distress syndrome, based on the idea that ACE2 is the SARS receptor and protects from lung injury. This molecule went already into 89 humans in phase 1, healthy volunteers, and phase 2 people with lung disease, ARDS, pulmonary hypertension, with very reasonably good safety profile. And then, of course, COVID-19 came about. And it was very clear, very fast, based on the sequence, that ACE2 must be and should be also a critical receptor. Of course, with all the other important things Stefan pointed out, which the virus needs to enter a cell. So all of a sudden, ACE2 went from being interesting, and nobody really cared about this, about SARS, to probably became the center of attention, a global center of attention, and probably has become one of the most widely researched proteins on the planet, from vaccine designers to AI companies, to small molecule predictions, to lots of research, and of course, with the idea to block this interface between ACE2 and the spike protein. Very rapidly, the structures were solved. I'm actually amazed how fast research has moved. Stefan and ourselves have been laboring away on this, and probably his experience is the same, and all of a sudden, thousands of groups have jumped into this stampede, which is of course great, because it has accelerated research massively, from companies to all the large universities and of course all research institutions which work on COVID-19. So this was the structure, cryostructure. On the top is ACE2. This is actually modeling we do ourselves. One thing I want to point out is the spike protein is heavily glycosylated. So beside ACE2, and of course people have published, there should be also lectins which could bind, and I think this will also be a very important mode of interactions, and how lectins and bindings of lectin to the spike would contribute to this. So recently we asked two questions. Could soluble ACE2 reduce the SARS coronavirus 2 infection, obviously being the receptor? The idea was really this, these two viruses use ACE2, bind to ACE2, of course furin and the other proteases come in to allow entry. The virus goes into the cells, uncoats, and of course does its thing, so damages cells, turns on the immune system, does other things, occupies the cellular machinery to replicate itself, causing diseases we call SARS and COVID-19. And so this was really the idea. If you have a soluble ACE2, which we had in clinical development for more than 10 years, it's basically a soluble human recombinant form. It lacks the transmembrane domain. It's more or less works like a neutralizing antibody with super high affinity, 1.2 nanomolar, like a very high affinity antibody. But of course, the virus cannot escape or mutate itself out from this like a monoclonal antibody recognition, because it mutates itself out from ACE2 recognition. It will not infect, or not infect at this level. So this was the idea. Maybe this would reduce the infection. So we hooked up with Ali Mirasimi in Stockholm in Karolinska. This is the virus he identified, I think, in the first Swedish patient with COVID-19. So this is actually the virus we use. And Ali's group in Stockholm infected Vero E6 cells with the virus. And then we asked the question if human recombinant protein, very similar what Stefan presented, with other molecules, would actually block infection. And indeed, APN1, this is actually the generic name for human recombinant soluble ACE2, can reduce viral load by a factor of 1,000 to 5,000 times, depending on the numbers of virus and, of course, on the dose of ACE2. We give mouse ACE2 does not do anything, as one would predict, because of actually doesn't bind. Mouse and rat ACE2, as we understand, does not bind to the spike protein. The second question we asked was really, which was critical, because is now COVID-19 a lung disease? And the second, and the manifestation in other tissues is because of the immune system is overshooting, or can other tissues be directly infected with the virus? And the reason was really because ACE2 is expressed all over the place. It's in the heart, in the blood vessels, in the nose epithelium, it's in the central nervous system. ACE2 expression changes with age, with gender. We actually mapped initially ACE2 onto the X chromosome. So that's why this, and in our animal models, we always saw gender bias. Expression changes with cardiovascular disease, with diabetes, smoking. And of course, it makes all sense because the renin angiotensin system is critical for all these systems. And of course, if there's counterbalance. We also published papers where we mapped ACE2 to the luminal surface of the gut epithelium, and also a paper where we mapped it to the proximal tubules of the kidney. So in the gut, it actually does something totally different. It stabilizes an amino acid transporter for neutral amino acids and controls dietary amino acid uptake. So basically, could ACE2 explain multi-organ COVID-19, not just by the immune system getting turned on, but by direct infection? And I think we all know now anyway that this is happening from autopsies and post-mortem analysis. But when we started, when it all the whole COVID-19 came about, we actually put together a consortium of tissue engineers in Europe and with virologists, with Ali Mirasimi's group, and basically engineered human kidneys, Nuria Montserrat in Barcelona. So her kidneys are very interesting because it actually has proximal tubules which express ACE2, which is because, just have an organoid, there might be different types of maturity. So if you use organoids for COVID-19 research and infection, we need to make sure ACE2 is expressed, and of course other molecules which facilitate entry into the cells are expressed. And different types of organoids might have different expression levels. To make it sure if we infect these kidney organoids, we can see viral RNA, so this replication of the viral, we see viral progeny. The virus actually makes new virus which can infect other cells. And we can again block with human recombinant ACE2 as you would expect. We also made gut organoids. ACE2 is expressed on the luminal surface. The virus can replicate. We never published this, but there are other groups which publish now viral replication gut organoids and many other organoids now. But this experiment I find very interesting. Last year, we actually engineered from stem cells blood vessel organoids, perfect capillaries with endothelium, pericyte, basal membrane. And so we asked the question if endothelium and blood vessels can be directly infected. And of course, as we know now, COVID-19 starts in the lung, in the nose, goes deep down into the lung. If it spills out, it can infect other tissues. But to infect them, it needs to go around in the blood and of course has to infect the blood vessels to get into the tissues. So to make it short, again, we can infect the blood vessels, we can find viral progeny, and again, we can block it with human recombinant ACE2. And there are all these reports now that COVID-19, at least in later stages, it's also a vascular disease, Kawasaki-like syndromes in kids, in adults, strokes, and of course, a link to thromboembolism. And of course, direct infection and of course, immunity and other factors which play in the response of the body to the virus can explain this on the endothelial cells. So based on all of this, we're actually now in phase 2 placebo double-blinded clinical studies in severe COVID-19 patients. So they already have severe disease, but not on intubation yet. The reason why we do this is because first, of course, it would work as an antiviral, very similar to remdesivir. And of course, we give it systemically. So unfortunately, we don't have it nebulized yet, but we give it systemically. And secondly, from data from my lab and many, many other laboratories, ACE2 protects against organ damage in multiple paradigms, heart disease, kidney disease, liver disease, cardiovascular diseases. Of course, ACE inhibitors are used all over the place for controlling cardiovascular diseases and diabetic nephropathies. And basically every time we knock out ACE2, these diseases get worse. And every time we replace ACE2 with an enzyme, we can make it better. What's important, these two functions are engineered in the same molecule and are independent of each other. So as far as we can tell, the virus binds on one side, but this does not affect catalytic activity and vice versa. So we have these two functions. We have started recruiting now in Europe and now talking to the FDA if we can expand into the US. So this is where we are. I can tell you, if you ask, we have a compassionate use patient, which is quite interesting because there's some issues which were very critical for us to address. For instance, if ACE2 binds to the virus, would this divert immunity? Would there still be an antibody response, a normal kinetics antibody response to the virus? So something weird is going on. And it actually does happen. The antibody response appears to be still normal. And we also see that the virus rapidly disappears in the blood and also later on in the lungs of the patient. So it seems what we are predicting works. Of course, now we need to do very careful clinical studies because as we all know from the experience, what's happening now, it's like this gold rush into developing medicines for COVID-19. Chloroquine was a classic example. But what we really need is carefully designed, controlled clinical studies. Because at the end, we need to come up with medicines which works, but not work by anecdote, but really work in many, many patients in controlled clinical trials. And of course, we hope ACE2 works, but of course, only at the end of the trial, we can tell. It might also not work, but at least we will get data to see if this is a therapeutic option, in addition, of course, what other people are doing. So with this, I end. Mike Krakauer identified ACE2 in my lab, made the first knockout mouse. Yumiko Imai was the driving force in setting up these lung failure models and finding ACE2 protects against lung injury. Rainer and Alex develop blood vessel organoids. Nuria, we collaborate from Barcelona. She does this beautiful kidney organoids. We also work with other groups which does organoids. And of course, this is not an exploding field using engineered human tissues for COVID-19 research, already pointed out by Darcy in her introduction, by many, many groups now in the world. And we do our infectious work with Ali Mirasimi. The person who actually does all the work is brilliant, is Vanessa Monte. With this, I end my talk and thanks for listening.
All right, so thank you, Joseph. There are already some questions in the chat, and I think both Joseph and Stefan hopefully can also see them. But I think one way I've been watching as you guys go, I think there's at least one question that maybe I can start with that seems to be brought up several times, is maybe you both could comment if there are other receptors other than ACE2 known or putative other viral entry receptors.
Maybe I can just go and then I'll go to Stefan. So I mean, I think we all agree ACE2 is very critical, which does not exclude there could be something else like lectins. I'm not personally not convinced by the data on CD147. If you actually go back to the primary paper, it's very, I'm not convinced by the data, but who knows? So there could be other receptors, but clearly ACE2 must be and is critical. Stefan.
Yeah, thank you very much. I actually have not much to add also from my perspective. ACE2 is critical. In fact, we just are assembling a small manuscript where we assessed potential attachment factors, including lectins and tyrosine kinases, and we see some effects, but none of those effects are so prominent that I would say they could be relevant in vivo. I saw that one of the participants asked regarding integrins, and this question addresses the presence of a potential integrin binding sites in the spike of SARS-CoV-2. We find if we mutate that, we see a very prominent phenotype, but we don't know whether this just kills expression. So we just have to do our immunoplots and additional controls. But this is all I know at present. So I would say, yes, ACE2 clearly is the major player in entry.
Okay, thank you to both of you. I think you guys can also see some of these. So I think some of these, I mean, Joseph, maybe you can answer quickly, but there's a question if your ACE2 knockout mouse is specific or if it's global.
It's global. We also made an ACE2 floxed mouse, which is, we never published, but we have it. And of course, like many others, we are now humanizing our mice. So we just should have the mice next week. So basically have a mouse ACE2 knockout with human ACE2. So we can use them, and of course we'll distribute them to people for infectious work.
Okay.
And we do floxed mice, and we're actually constructing now an allele where we can knock out mouse ACE2, for instance, in endothelial cells or in lung cells or in brain cells, and then only in this cell type put in human ACE2. So to really see, you know, what would actually happen in different tissues.
Okay, I think the next question that just came up here at the end is if you guys, I think Joseph, you made a comment on ACE2 expression and aging.
Yeah, there's some papers coming out that ACE2 expression in children is much lower than, and it's you know, they advance in puberty, then ACE2 expression gets increased in the respiratory system. There was also data coming out that in kids, ACE2 expression in the gut is lower and it increases with aging. Okay. But I think we have to be very careful with this ACE2 expression data at the moment because there's some bad reagents out there. There's some bad papers out there making claims which is probably not real, but based on, you know, bad antibodies. So I think
could you maybe spare a lot of people some work? If there's antibodies that you guys have found in your hands, if you could make some recommendations for people that are, again, trying to just get into this field to begin with, save them troubleshooting.
You know, to recommend now a certain company online might not be very wise for me, but I think we might actually get together all and, you know, and actually make recommendations which antibodies work or not. I think this would be really beneficial.
Maybe I can ask the question a different way. Have you been transparent in your papers about the methods with the antibodies that you have used, so people could reference the papers and then look at concentrations, for example, and conditions, you know, that can be used based on your published work?
Always transparent, of course.
Yes. So then that answers that question. So people can go look at your papers and they can use those antibodies, which you feel that have been well validated and confirmed. So I think there were some, before we get to the newer questions that are here at the bottom, it seems that there's some other questions about, and I know that I've also seen some presentations on potential connections between interferon gamma signaling and ACE2, and it's in the context of its function in the Renin angiotensin system. I don't know if you can comment on that, Joseph.
Yeah, there's this data interferon turns on ACE2 expression. Stefan, does this also turn on expression of the proteases? Or?
Thank you. That is a good question. Actually, we tried this, but not very in depth, and we don't have evidence that TMPRSS2 expression is induced by interferon, but we didn't put too much effort into that, so I can't really give a definite answer.
Okay. Let's see. So there's a question here at the bottom, which I think you've addressed, but maybe you guys can comment. So which cell type will be a better model to study cell entry and viral replication inhibition to mimic the in vivo process? So I think, Stefan, you showed some data on that. So maybe you want to just, you know, confirm that.
Thank you. Yes, sure. So from our perspective, particularly looking at the chloroquine, hydroxychloroquine thing, Vero E6 cells are not a good model to study viral entry. You want the lung cell line, which has the major entry factors, which are ACE2 from our perspective, and TMPRSS2. So we are frequently using KLO3 cells and feel that they are at least a reasonable good model. Of course, not primary cells, just the cell line, but at least receptor expression is good or mimics what is seen in the lung.
Okay, so I'll maybe ask another question, maybe if other audience members are thinking. So in addition to sort of viral entry, we know from a lot of patients that it's really maybe the aberrant immune response that is actually in the end what causes mortality. So not viral replication per se, but the response. So I'm just wondering if you guys are analyzing sort of cytokine responses and any of your model to sort of add to that literature of how we can understand what this virus really does to the immune system. So independent sort of a viral entry per se, but what it does. Yeah, so I don't know if either of you want to comment on that.
I give it over to Stefan, he's the virologist.
Thank you very much. That's far too much honor. So I'm afraid I cannot really contribute on that topic. I'm unfortunately a virologist and not an immunologist. However, I would like to make shamelessly some advertisement for our strategy, because I noted with some interest that one of the JV papers which assessed the role of TMPRSS2 for coronavirus infection, it found that it's required in mice. That is good. But it also reported that upon poly-IC treatment, the release of pro-inflammatory cytokines is reduced in that mice. So we think that there's a certain possibility that our protease inhibitors not only have direct antiviral function by blocking the virus activating protease TMPRSS2, but also by maybe reducing cytokine release. That is a hypothesis that we will for sure pursue. But unfortunately, I cannot add more.
Okay.
I mean, one
Joseph, do
thing
you have
from
anything?
ACE2, yeah, one thing from ACE2 I can add, angiotensin 2 is known to drive inflammation. So there are actually clinical studies where people infused patients with very low blood pressure and critical care patients with Ang2. And basically what happens is thromboembolism is one of the major side effects, which is really interesting. And IL-6 goes up, for instance. So it has been known for many systems that Ang2 is pro-inflammatory. So we also believe ACE2 should at least contribute in part to reducing inflammation, which we actually do see in our compassionate use trial, that inflammatory cytokines go down. But again, you know, we need large trials to confirm this.
Yeah, and then maybe I'll ask another question while I wrap my head around this other one. So maybe you guys can comment a bit on the timing of when these agents are given. So again, a patient presents symptoms, they're presumably already then quite infected at that point. So is it really then just a matter of, you know, again, preventing further viral replication while the natural, you know, immune system is able to sort of clear the virus that is already there? So just maybe if you can comment on sort of the timing, or if it's when a patient really presents with symptoms that, you know, we should be giving drugs, you know, in case they advance. So I don't know if you guys can comment on sort of the clinical trials that you're aware of what the timing is of when these agents are being given. Yeah,
in our study, we do intravenous infusions. So we set up the study actually at a later stage of the disease, stage 2B. You know, we could make a very good argument to go actually much earlier, but we had to focus on one patient population, because if you go earlier, then you have to have thousands of patients. So now we'll include 200 patients. So yes, and it would be also very interesting to nebulize ACE2 or ACE2 spike interaction blockers and see if one could prevent or protect primary caregivers or, you know, or in a hospital setting. So, but we are not there yet. So we had to focus on the late stage.
I think that probably Joseph will agree. The general idea is that if you come in with an antiviral agent, you want to come in as early as possible, which for technical reasons as has been said, is not always or frequently not possible, but that would be desirable. So if you have first symptoms, if you're in a risk group, you should be treated in an ideal world, but it's not an ideal world.
Okay, so maybe I'll ask one final question here, which appeared twice. So it says ectodomain shedding of ACE2 has been associated with infections by viruses. How does that relate to SARS-CoV-2 infection?
I don't know. To be honest, we don't see so much shedding because we have actually set up very sensitive systems with mass spec, where we can read out the RAS system much more sensitive what's actually going on in the serum. And we don't see much of shed ACE2, at least in the system, which does not mean it's not important and not contributing, but I don't know.
Stefan, do you have any further comments on that?
No, I don't.
Okay, all right. So with that, we are actually out of time. So I want to thank both of our two speakers for excellent presentations and also for taking the time out of your really busy schedules. I know that for all of us that are doing COVID-19 research, that our worlds have been turned upside down. So again, we really, really appreciate you taking the time for the ERS webinar for today. We will share slides and information for everybody else that's still here so that you can get this information from us. So thank you again for attending. And I wish everybody luck in their research and hopefully you're enjoying the summer or the winter, wherever you're living right now, despite the pandemic. So thanks again. And yeah, hope to see all of you soon in real life somewhere. So bye.
Thank
you very much.
Good night.
Yep.
And good day. Bye.